Okay, so this is the regulation of gene expression in prokaryotes lecture, and we will be beginning with prokaryotes. So the first part of the lecture will be looking at regulation of gene expression in prokaryotes. We'll look at sort of an overview of some of the general gene regulatory mechanisms, some of the terms that we need to know in order to understand gene regulation. And then we will take a specific look at one case in particular, and that is the lac operon. In the second part of the lecture, we'll be looking at regulation of gene expression in eukaryotes, which, as you might imagine, is far more complex than regulation in prokaryotes. So we'll look at some of the aspects of this, including chromatin structure and dynamics, the role of nuclear architecture, etc. So to begin with prokaryotes, one environment that we have literally billions of bacteria growing is our gut. So this is a scanning electron micrograph of some of the bacteria that can be found in our gut. And these bacteria are constantly responding to changes in the environment. So they have to quickly adjust their gene expression profile depending on which nutrients are available to them. So in order to change their gene expression to adjust to the environment, they have to control this genetic flow. So in other words, they have to control which genes are turned on to make protein. And there are several places at which they can actually control this process. So as you might imagine, they can control the very first process, which is the process of converting DNA of a particular gene into a messenger RNA. So this is the regulation of transcription. In addition to controlling transcription rates, etc., cells can also control how quickly this messenger RNA is translated into protein. So this is, of course, regulation of the translation step. In addition to controlling those two processes, there's also ways that the cell can put modifications on this final protein in order to modify its activity or its stability. And those types of regulations are called post-translational regulation. It turns out that there's something like 200 different modifications that can be made on a protein to alter its activity. So for a second, I'd like you to think about which points of regulation here would be the quickest changes that you could make if you wanted to alter gene expression, and which points of regulation would take more time. So if you want to quickly regulate a protein, the fastest way to do this is actually to modify the protein itself. Okay, so you can quickly, rapidly slap a phosphate group or add a modification group to a protein and alter its activity, and even cause that protein to be degraded. So this is the fastest place that you can regulate gene expression. However, because you've gone through all of this work to get that gene transcribed and translated, this is energetically expensive. It wastes a lot of energy when you're just modifying the protein. If instead you regulate the beginning of this process by regulating the rate of transcription, this is energetically favorable. This is going to save you all of that energy that you would have put in to transcribe and translate that protein. However, this is going to be slower. You've got to go and turn off all of the transcription factors so that they're not binding to the promoters, etc. Okay. So with this in mind, this is sort of a framework for the ways that we'll look at how prokaryotes and eukaryotes can regulate gene expression. Today we'll mostly be focusing on this first step, this regulation of transcription, which, as we've mentioned, is slower, but is also the most energetically favorable for the cell. Before we get started into the main meat of the lecture, we do have to go over a few terms for gene regulation. So this is some of our gene regulation terminology. Today we're going to be talking a lot about different trans-acting elements. And these trans-acting elements are generally proteins that are proteins such as the ones shown here that are going to be binding to regions of the DNA, typically upstream of a particular gene, to regulate that gene expression, okay, that transcription event. These are considered transacting because they are expressed typically from other locations in the genome and they act in trans on a particular gene. The cis-acting elements are typically DNA sequences that are right upstream of a particular gene, and these are the regulatory sequences that are bound by these transacting elements. If we look at some examples of transacting elements that we'll be talking about, some of these you'll see are considered activators. Activators, as their name implies, are going to be positive regulators, or in other words, proteins that are going to be increasing transcription. So they're binding to cis-acting elements and increasing the rates of transcription. We'll also talk about some examples of repressors, which, as their name implies, are negative regulators of gene expression. So they help to decrease transcription. Some of the cis-acting elements that we'll be going through today include regions upstream of the gene, including the promoter sequences. As we've discussed in previous lectures, the promoter sequences are typically bound by a transcription factor. In the case of prokaryotes, this could be sigma, which brings in the RNA polymerase. Or if we're talking about eukaryotes, this would be the general transcription factors that would bind to the promoter. There's other types of cis-acting elements or DNA sequences, including the operator. This one will be important for the lac operon. This is a particular DNA sequence that is bound by a transacting element called a repressor. And we'll look more into that when we talk about the lac operon. There's also sites that can be bound by activators. And so in the case of the lac operon, in addition to a promoter and an operator, we'll also look at an activator binding site. And this is going to be the site at which one of the activators binds. A couple of other terms that we'll need to know for this gene expression lecture are constitutive and inducible. So constitutive, another word for constitutive is constant. So we'll look at some genes that are going to be constitutively expressed. This means that these genes are turned on all the time. So what kind of genes would you think would be turned on all the time in a cell? So you might be guessing maybe housekeeping genes, genes like some of the metabolic enzymes, genes that encode these metabolic enzymes. These enzymes have to be expressed all of the time in order to carry out the normal work of the cell. Inducible genes are genes that can be turned on or off depending on the environment of the cell and the needs of the cell. So can you think of any examples of inducible genes? One example that we'll talk about today is the genes that are encoded by the lac operon. These genes encode enzymes that break down lactose. And we'll see that the cell doesn't always need to express them. It only needs to express them when there's lactose around to use as an energy source.